The air in the basement of King's College London tastes of ozone and stale coffee. It is May 1952, and the world outside is rebuilding from the ashes of war, vibrating with the early hum of the atomic age. But down here, in this subterranean concrete box, time seems to have stopped. There is no sunlight. There is only the low, menacing buzz of the X-ray diffraction machine and the rhythmic breathing of the woman standing in front of it. Rosalind Franklin. She is 31 years old, her dark hair pulled back efficiently, her eyes dark and burning with a terrifying intelligence. She is not here to make friends. She is here to force the invisible to show its face. She is attempting something that every other physicist in Europe has quietly decided is impossible. She wants to take a picture of life itself. Not a cell, not a nucleus, but the molecule inside it. Deoxyribonucleic acid, DNA. At this moment, DNA is a ghost story in the scientific community. They know it exists, they suspect it carries the code of heredity, but nobody knows what it looks like. It is a chaotic, wet, fragile mess. If you look at it wrong, it collapses. If you dry it out, its structure shatters like antique glass. To photograph it, you have to bombard it with lethal radiation while keeping it alive in a perfectly simulated artificial atmosphere. Rosalind leans over the camera assembly, her face illuminated by the eerie blue glow of the X-ray tube. She is adjusting a single fiber of DNA, thinner than a human hair, suspended by a glass rod. This is not standard photography. There is no lens, there is no shutter click. This is diffraction. She has to shoot X-rays through the crystal and catch the scattered shadow they cast on a piece of film behind it. It is like trying to determine the shape of a chandelier by looking at the shadow it casts on the floor while swinging in the DAC. The technical challenge is a nightmare of physics and chemistry. The DNA fiber demands precisely 75% relative humidity. Any less, and the A-form crystal structure dominates, dry and useless. Any more, and it dissolves into a shapeless gel. She has spent months designing a tilting camera chamber that seals hermetically, bubbling hydrogen gas through salt solutions to maintain that equilibrium. She is not just a biologist, she is an architect of microclimates. She works with the hands of a bomb disposal expert. One slip of the tweezers, one fluctuation in temperature, and weeks of preparation are ruined. The machine begins its work. The exposure time is not a fraction of a second. It is not minutes. It is 100 hours. 100 hours of continuous, high-intensity X-ray bombardment. For 4 days and nights, the machine hums its deadly lullaby. Rosalind does not sleep. She naps in a chair in the hallway. She checks the gauges. She monitors the gas flow.
She is alone in a male-dominated department that views her with suspicion. A woman who doesn't smile, who doesn't defer, who corrects their math in staff meetings. They call her Rosie behind her back, a diminutive nickname she detests. To them, she is difficult. To history, she is about to become the most important photographer who ever lived. The radiation is everywhere. In 1952, safety protocols are primitive. She wears a badge to measure exposure, but she often forgets it or ignores it. She stands in the beam's path to adjust the collimator. She manipulates the crystals with her bare hands. The invisible fire that allows her to see the secrets of life is slowly, silently rewriting the code of her own cells. She is trading her future for this image. Finally, the 100th hour strikes. The hum stops. The silence in the basement is deafening. With trembling hands, she removes the film cassette. This is the moment of truth. She takes it into the darkroom. The smell of acetic acid and fixer fills the small, red-lit space. She slides the film into the developer tray. She rocks it gently. The fluid washes over the gray sheet, and slowly, like a spirit manifesting from the ether, the pattern emerges. It is sharp. It is unmistakable. A heavy black cross of reflections dominating the center of the image. The spots are arranged in a perfect symmetry that screams out from the grain. To the untrained eye, it looks like a blurry X, but to Rosalind Franklin, it is a shout. It says helix. It says 2 strands. It says the phosphorus are on the outside. It is photo 51. She holds the dripping film up to the safe light. It is the Mona Lisa of biology. It is the answer to the question humans have asked since they first looked in a mirror. What are we? The data is all there. The spacing of the bars tells her the pitch of the helix. The missing reflections tell her about the symmetry. She doesn't run into the hallway screaming Eureka. That is not her way. She is a scientist of the highest rigor. She dries the film. She places it in a folder. She locks the folder in her desk. She intends to calculate the Patterson function, to prove the structure mathematically before she speculates. She believes she has time. She believes the data belongs to her because she earned it with her sweat and her blood. She is wrong.
The atmosphere at King's College has curdled into toxic silence. The corridor between Rosalind Franklin's office and that of her colleague, Maurice Wilkins, might as well be a minefield. They are supposed to be partners, but they are barely speaking. Wilkins, a quiet, socially awkward man who expected Franklin to be his assistant rather than his equal, is shrinking under her intensity. He cannot handle her directness, her terrifying competence. He retreats into the shadows, nursing a growing resentment. He feels locked out of his own lab, excluded from the data that he feels, by virtue of seniority and gender, should be shared with him. Meanwhile, 70 miles away at the Cavendish Laboratory in Cambridge, two other men are growing desperate. James Watson, a brash 24-year-old American prodigy with untamed hair and an arrogance that barely fits through the door, and Francis Crick, a loud, boisterous talker with a mind like a relentless drill. They are building models. They are playing with cardboard cutouts and metal plates, trying to guess the structure of DNA without doing the hard experimental work. And they are failing. Their models are embarrassing. They have put the sugar phosphate backbone on the inside. They have proposed 3 strands instead of 2. They are grasping at straws, trying to solve a 3-dimensional jigsaw puzzle with half the pieces missing and the lights turned off. The head of their lab has ordered them to stop, to cease their DNA work because they are embarrassing the university. They are stuck. They know the prize is within reach, the Nobel, the fame, the answer to everything, but it is slipping through their fingers. They need a miracle. They need a map. On January 30, 1953, the timeline of history bends. James Watson takes a train to London. He heads to King's College, ostensibly to visit Wilkins, but really to sniff around. He runs into Franklin first. He barges into her lab, interrupting her work, and begins to lecture her on why her interpretation of the data is wrong. He patronizes her. The encounter turns volatile. Franklin, pushed to the breaking point by months of condescension, advances on him. Watson, startled by her fury, retreats. He backs out of the room, fleeing the dark lady. He runs straight into the arms of Maurice Wilkins. Wilkins comforts the shaken American. She's impossible, they agree. She's hoarding the data. In this moment of shared grievance, the bond of the old boys club tightens. Wilkins wants to show Watson that he is not crazy, that there is real data, that he is in the loop. He invites Watson into a side room. He walks over to a file cabinet. Wilkins' hand hovers over the drawer handle, a mundane action that will echo through eternity. He pulls it open. He takes out a print. It is photo 51. Franklin is not in the room. She has not given permission. She does not know this is happening. This is her work, her months of radiation exposure, her intellectual property. And in the casual camaraderie of 2 men bonding over a difficult woman, it is handed over. Wilkins holds the photo up for Watson to see. The reaction is instantaneous, visceral, and violent. Watson later wrote in his own memoir, "My jaw fell open and my pulse began to race." It takes him less than 10 seconds. That is all. In 10 seconds, the genius of Rosalind Franklin's technical perfection transmits the truth to James Watson's prepared mind. He doesn't need to do the math. He sees the black cross. He sees the reflection on the meridian. The image is so perfect, so clear, that the structure leaps off the page and punches him in the face. It's a helix. It creates a complete revolution every 34 angstroms. He struggles to keep his face neutral, to not give away how much he has seen. He thanks Wilkins. He rushes out of the building. He is practically vibrating. The train ride back to Cambridge is a blur of adrenaline. He sketches on the margin of a newspaper. He knows. He knows the secret. Back at the Cavendish, he bursts in on Crick. "It's a helix," he announces. They don't have to guess anymore. They have the dimensions. They have the parameters. They clear off the tables. They call the machine shop to cut new metal plates. They are not running experiments to test a hypothesis. They are reverse engineering a solution from the stolen answer key. Watson and Crick feverishly assembling the metal model, tightening bolts, the structure rising like a golden spiral tower. They work with the manic energy of thieves who know the police are closing in. They twist the bases. They align the bonds. Suddenly, the cardboard pieces fit. Adenine pairs with thymine.
Guanine pairs with cytosine. The structure locks into place. It is elegant. It is beautiful. It is perfect. And it is entirely built on the foundation of the photograph lying in a dark drawer in London, taken by a woman who is currently sitting alone, analyzing her data, unaware that her life's work has just been claimed by someone else.
The announcement hits the world like a thunderclap. In April 1953, Nature publishes 3 papers. The first is by Watson and Crick, proposing the double helix structure of DNA. It is a masterpiece of deductive reasoning, a short, punchy article that changes biology forever. The second paper is by Wilkins. The third, buried at the back, is by Rosalind Franklin and her student, Raymond Gosling. To the casual reader, it looks like a collaborative triumph. Watson and Crick propose a model, Franklin provides the supporting data. But the chronology is a lie. The model was built from the data, not confirmed by it. In their paper, Watson and Crick offer only a dismissive, cryptic footnote acknowledging that they were stimulated by a knowledge of the general nature of the unpublished experimental results and ideas of Dr. M. H. Wilkins and Dr. R. E. Franklin. It is a masterclass in minimization, stimulated by, not dependent on, not impossible without. They have taken the brick she baked and used it as the keystone of their arch, then thanked her for the clay. Rosalind does not fight them. She does not know the extent of the theft. She assumes they deduced it independently or saw an earlier report. And she is tired. She is done with the toxicity of King's College. She packs up her files. She moves to Birkbeck College, leaving the DNA work behind. She is a scientist, not a celebrity. She moves on to the structure of the tobacco mosaic virus and later the polio virus. She does brilliant, groundbreaking work. She travels. She climbs mountains. She laughs with friends who know a different side of her than the dark lady myth constructed by Watson. But the clock is ticking. The radiation, the invisible fire she courted for so many hours in that basement, has done its work. The double-stranded breaks in her own DNA, the irony of which is too cruel to contemplate, begin to manifest. She falls ill, ovarian cancer. The pain is excruciating. She works through it. She hides her illness, crawling up the stairs to her lab when the elevators are broken, refusing to stop until her body simply cannot continue. Rosalind working in her final days, pale and frail, surrounded by virus models, the light fading from the window. She dies on April 16, 1958. She is 37 years old. Her obituary celebrates her work on viruses. DNA is mentioned as a footnote. 4 years later, in 1962, the call comes from Stockholm. The Nobel Prize in Physiology or Medicine is awarded for the discovery of the molecular structure of nucleic acids. The winners are James Watson, Francis Crick, and Maurice Wilkins. 3 men stand on the stage in white ties and tails. They bow to the King of Sweden. They accept the applause of the world.